Hi, thank you everyone for coming here, and thank you to the organizers for giving me a chance to present my work and putting me on the same day as Gunnar. So please don't judge Swedish science by what I say, wait till the end. So I'll be talking to you about transmembrane beta barrel proteins. They are one kind of membrane protein, so I'll talk to you about how we predict their topology and how we get their structure out. So generally when there are talks on membrane proteins, you see these two structures, alpha helical ones on your right-hand side, and then the yellow ones on beta barrels. And then generally the presenter says, now we will talk about beta barrels. So, well, they say about helices, now we'll talk about beta barrels here. The brief outline is as follows. So I'll try to give you a rough idea about how their structure looks like. Then why they're important. I mean, there are a few groups working on it, but there are a few more coming up. So it will be interesting to find out why we want to work on them. And then I'll discuss some results based on our two publications. One is WalkTopus, which is where we get the topologies from, and then the 3D modeling using top model. So what you see in the figure is the rough outline of what I want to develop as a framework. So I want to start from a protein sequence, then using different methods, you can predict their topology, and then you have to arrange these strands. So you can arrange them by moving up and down or changing the angle, and then you can get a kind of a barrel. You have to arrange them, and you can get a barrel. So what I'm interested in is redesigning them or getting membrane protein complexes. So far we limit ourselves to the membrane region, because if you get the beta strands right, then more or less you can do both these things. All right. So this is a rough kind of a structure. Well, this is a real structure, but like this is what we use as a representative structure here. So you have the inner loops on the periplasmic side. The outer loops are on the extracellular side. Yeah. What is quite common is that the outer loops are really, really big, and the inner loops are really small. And at least in the bacteria, the beta strands are anti-parallel, and they're even in number. But if you go to mitochondria, there's one example, one structure available where it's odd. There are more complex examples that I'll come to, but so far, this is what we are using as our reference structure. This is the top view. So if you look at the barrel, if the barrel is nice, then you can predict nicely the strands. But if it's not nice, and it has all these strands within the barrel, which make your prediction difficult, but I guess they are functionally very important. All right. So as I said, that the reference structure that we have is a very basic beta barrel. But if we go and see the complexity, we can have all different kinds of protein chains coming together and forming beta barrels. So you have the simplest one here, and I guess the yellow color was not a good choice, so you don't see the plug domain that is somewhere in there. Then you have a huge helix that goes through the barrel. Then you have these multi-chain ones where they are homotrimers, or 7 chains come together to form a barrel. They're all in gram-negative ones. And we have some examples also from gram-positive ones where they are all multi-chains, and they work as toxins and pathogens. So they're medically also relevant. I mean, I was thinking that they're statistically very similar because, I mean, they are all beta strands. But I mean, I guess then all proteins are statistically similar, but biologically they're different in the sense that their insertion mechanism is different. Another thing that is important, why we have to work on them, or why it's important to work on them, is the translocation machinery. So both in bacteria and mitochondria, and as well as in chloroplasts that I'm not showing here, the central machinery that is responsible for the insertion. So here, this green YAET, the BAM complex here that you have, and here the TOM complex, they are both beta barrels. And the TOM complex structure is not known. So it's kind of relevant to come up with methods with which we can predict their structures. And another, just one more final motivation is just kind of special and special interest for me is that they are responsible for one of the secretion systems, the type 5 secretion system, and that can be further subdivided into TPS, which is a 2-partner system, and then the autotransporters. And they're both related to somehow diseases. They're not very fancy diseases like Alzheimer's, but still they are diseases which are very prevalent in developing countries. All right. So with that in mind, I'll just directly go to the results. So what we want to do is, first of all, from the sequence, we want to extract a topology. How we do it is that we take the sequence. It's a two-stage process, first of all. We have support vector machines in the first stage, and in the second stage, we have the hidden Markov models. So how we do it is that we take the sequence, run it using HH blades, get sequences out, generate a position-specific scoring matrix, and then with that, we test and train 3 different support vector machines. The 3 ones are for predicting the I region, the O, and the M. So the O is the outer loops, M is the membrane region, and I is the inner loops. So once we get these probabilities out of a residue to be in a certain location, we combine the probabilities into a profile and feed it to an HMM. So we do that so that we can also take into account the global preferences of the residues and get better accuracy, of course. So the architecture of the HMM is shown here. It's one-to-one. We try to get it one-to-one correspondence to the reference structure that we have. So we start in the inner cytoplasmic side and also end in the cytoplasmic side. We have an inner loop here, which is we haven't limited the state, the maximum size. Then we have the strand, so you go up, then you go to the outer loop, and then you come down. So the HMM is based on the package developed previously, I think with Arne, mod HMM. So all the transition probabilities and emission probabilities in a given state are set to 1, except the ones which are the loops here. So these are the ones that we had to optimize. And so while we were optimizing them in a very strict 10-fold cross-validation, so we start from the sequence until the end, we maintain the training sets and the test sets separately. While we were optimizing them, we got a lot of different sets of parameters which gave us similar results. So since the data set is only 36 proteins, it's quite reasonable that we get various parameters that give us similar results. All right. So if we just have the SVM, so on the y-axis you have the MCC, and on the x-axis you have different window sizes. So if you just have the SVM, this is the kind of accuracies that you can get. For the membrane region is around 0.8, which is quite good. And then for the outer loop, it's around 0.75, and for the inner loop, it's a bit lower. The inner loop is a bit difficult to predict here because they're really short, and you have to optimize a lot the SVMs to get it right. But the good thing is that we can get the membrane region out correctly. Now we have a new data set, and there we divide this membrane region into in-facing and out-facing. So if you have a barrel, you can have the residues which are pointing towards the pore or towards the lipids. And surprisingly, this accuracy is quite high. Well, these are the good results, but still, there are still problems. So we get topologies out of it, and then we compare it to different methods that are available. So our results are based on this 10-fold cross-validation. So this is, I guess, the worst that we can do. So we can get 30 out of 36 number of strands correctly. I mean, number of strands is one measure of getting your accuracy measure. The other one would be getting the topology right. So that would mean that you have the number of strands right and also their location right. That's roughly 25, which is the same as Prof TMB. But I guess the results that we got from other methods are from their web server, so it's plus minus 1 or 2 maybe. So the other interesting thing is that the publications, they all are 2004, 6, 8, 10, 12. Well, there are some others, but they were not performing that well. So there are not many topology predictors available for beta barrels. All right. So once you have the topology, you could go through the whole set of sequences and try to combine this topology measure as a filter for getting beta barrels out of a given set of sequences. So what you can do is, I mean, what we tried is very simple, that if you predict 8 or more than 8 number of strands and you combine it with available methods, can you get the results a bit better? And indeed, we can do that. How that happens is that when we use this additional pre-filter kind of module, we reduce the number of false positives. So you take your favorite method, and then if you can use this Boctopus topology prediction there, then we increase the accuracy in all cases. All right, but I mean that's not really the aim of the talk. So we have published it, and then the web server is online. It usually works fine, and what you get as an output is you get the support vector machine's probabilities. So blue is out loop, I is in loop, M is the membrane region in gray, I guess. And then you also get this topology. You also get the text output, but it's good to visualize things. All right. But there's another thing to it. So you have all these parameters. So we decided, let's say we have all these parameters, why not we just test all of them? So we ran all different combinations of parameters 10,000 times, and then we had some errors. So we wanted to see if we can change, like, get a correct topology somehow. So for example, for this one, 3CSL, this is the percentage of results that are incorrect. So this one we get incorrect all the time, and some of them we get correct like 35% of the time. The errors that occur are divided in two parts. So you can have the overlap error, so where basically you get the number of strands correct, but they don't overlap correctly. So for example here, in the second example, let's say here, the strand is predicted a bit further away. And the other kind of errors that you can have is when you predict the wrong number of strands. One thing that we noted here was that most of the errors, or quite a few of them, were occurring at the pre-barrel state. So pre-barrel is somehow a state which is before you get to the barrel state. So you have the sequence, and the barrel is somehow at the end, and then before that you have a pre-sequence kind of thing. And that could be a whole protein in itself, I guess. And that's where we are making these errors. So just to have a kind of a visualization what I'm talking about. So if you have a beta barrel, it could have a whole kind of a plug domain inside it, which could be as big as 130 residues and can also consist of beta strands. And somehow that's where we make mistakes. And they can be even more interesting. So they could be even like 355 residues. They could form a helix which goes through the barrel and then have a whole structure on its own. So now we have extended the data set a bit. So this is on 45 proteins. So roughly half of them have this pre-barrel state, which is roughly 25 to 30 residues or even longer, and they can be like 300, 400 residues long. Okay, so we still have that error. Keeping that in mind, what we also wanted to have is a 3D representation of our topologies. And we were hoping that with the 3D topologies, we would be able to get rid of some more errors. So we developed a pipeline, which is as follows. So you have the protein sequence here. You can generate these multiple topologies, alternate topologies using different parameters, using Boctopus. And then you generate multiple C-alpha 3D models. So how we generate these is it's independent of a template model. So it's a mathematical. These are some old mathematical equations that are found, and we just use those. So it's a really fast method. So you can do this step very fast, and you generate lots of model very quickly. And then once you have a model, you can get the z coordinate of each residue in the membrane region. So the z coordinate is defined as so you find the membrane center, and then from that membrane center the distance of a residue. So that's one way of getting z coordinate. So you have lots of models, you get lots of z coordinate profiles. The other way would be you take the protein sequence and you train another support vector machine or support for regression, and then you develop something called ZPRED3, which is an extension of ZPRED that RNA has for alpha helical proteins. And then you just compare those two z coordinate values, and you can rank your models. So this is a very simple pipeline. Of course, in the future, it can be made more complicated and more robust, but this is what we have now. Just one slide on this z coordinate predictor. So we tried different parameters. So on x-axis, you see PSSM, the predicted topology, and all these things. And what we chose out of again a tenfold cross-validation is PSSM with the predicted topology. It gives us the highest percentage of residues within 2 angstrom of the observed value and has the lowest average error. So how do we actually do the modeling? So an ideal barrel can be defined using 5 parameters, which was discussed in 1994. So the parameters are the tilt. So you have a strand and you can like tilt it normal from the membrane normal. Then you can calculate the radius based on the number of strands. So n is the number of strands, a is the inter-residue distance, and b is the inter-strand distance. So you can calculate tilt, you can calculate radius based on it, and then you can calculate the tilt and coil of the beta strand. This we have not done so far. We have done the tilt and the radius, and we try different strand numbers, and that's how we generate a lot of models. So you try different strands, different shear numbers, and you can get different values out of it, and then you get different models. Then once you have that was the way to get strands, and you still have to arrange them in a kind of an ideal barrel. And that's again a very old paper, 1984. So we just simply transform our coordinates from x, y, z to something like FGH coordinates. So it's just a formulation where the centrals, so we choose one strand, and that is the center axis. So once we've done that, we again try to compare if we've done any better with the topology prediction. So, well, that's one of the results that we get. So if we would have done this 10,000 times, this again is like this is a reverse result, kind of like you have the correct topology. So here you don't predict any topology correct. So that we can't change, because if the topology is anyway wrong, then no modeling would in this way will help you. But if you have where you have some small percentage of correct topologies within the predicted topologies, that we could extract. So out of the 20 that had some sort of an error, we could extract 11 correctly. So if we count blue, so I've divided them in overlap and the number of strands incorrect. So this comes out to be 20, and then you reduce the error from 20 to 9. Again, the error somehow is a bit more nicely distributed now. Well, so still in 4 cases. So 1 E54, 3 CSL, 1 KMP, and 2 IAH, the error is still in the pre-barrel state. So yeah, one thing that we are currently working on is getting a predictor or some sort of a method correct for this pre-barrel filter. So I should show some models then, because it's about modeling. So these are just the 2 examples. Like we could get up to 3 angstroms, and again, we can also, this just shows that Bok-Topos predicted 51% of the time a wrong topology, but with top model we can extract the correct one. And in this one, it was 80% of the time this wrong topology, but we can still extract the correct topology out of it. So that's pretty much my final slide. So we have also compared it to the only 2 other methods that are available. One is quite recent, which came almost the same time as us, is this Jack's paper in 2012. And then this TMB Pro from Professor Baldi's lab in 2008. So, so far, I guess we are the only method that we can start from the sequence and go to the model. The TMB Pro model, the TMB Pro method makes some errors in the topology prediction. So if you have got the topology wrong, then of course you get the models wrong at the end. If we get the topology right, which is shown here in the last line, and on average we make an error of 5.8, and so this is on the data set of the method called 3D spot, not on our data set, and but so they're still a bit better, and I guess these 2 methods are really good, TMB Pro and 3D spot, because they more focus on hydrogen. So for example, 3D spot focuses more on hydrogen bonding and side chain interactions, and TMB Pro combines different methods together to get a 3D model. So it will be very interesting to combine these ideas together with a simple approach that we have of just using the Z coordinate prediction as a ranking scheme. And with that, I would like to conclude. So We have a Boctopus with which we can predict the topology. We've set up a web server for that. Then we have ZPRED3 with which we can predict the Z coordinates. And then we combine both of them and make another method which can rank 3D models of TMBs. The work is still continuing, and we are making it a bit more robust, and yeah, it's still far from finished. I would like to thank Professor Arne Olofsen for giving me the opportunity to work on transmembrane beta barrel proteins. Christoph Peters, he's a PhD student in our lab, and we are working together on improving beta barrels and topologies, and our collaborators, we have a lot of discussions with them. And I would like to also thank ISMB 2009, where I met Arne, and I got my postdoc there. And thanks a lot for your attention. Thank you.
Thank you for a very good, very interesting talk. If there are questions, yeah, maybe go to the mic. And in the meantime, I'll ask a quick question. So, I see that you are testing various ways along the way until you reach the final machine learning tool that you use. Does that require correction for multiple testing? You realize that you're testing various options to get the best result, and that, like I wonder if statistically that means that you have to correct for multiple testing.
Yeah. Sure. Yeah. I mean, one thing that we could look into is so our data set is not balanced. So that's what we are focusing on now, basically. So we change different parameters based on how we can have equal data sets in both bins. So now we are testing other other machine learning algos. That's what we are trying to do. I don't know if that's what you mean, but maybe.
Probably not, but we can talk about it later. A question there?
Yeah. You fed BioOctopus only with the structures or with the sequences of the structures that were available?
Oh, sorry, I forgot to mention that, I guess. So we have a data set of 36 proteins, which is reduced at 30% sequence identity. So there are 36 structures available. Well, there are more structures available, but there are 36 structures that we use to train our data.
Did you think about, instead of just using the sequences of these structures, to additionally complement this with highly similar sequences so that you boost your kind of sequence data and capture more of the variability that is out there and get somehow the more conserved residues to play a greater role in the SVM?
Yeah. Yes. So we haven't looked into that in that regard, but what we have tried is these new methods like Sykov and Professor Sander's contact predictor, and they seem to not work because these are not kind of contacts that. So these are more like backbone contacts. But yeah, we could look into just using the sequences to get more information. Thank you.
Yeah, hi, very interesting, and I hope we can talk offline. I just have some background question. Maybe I apologize if you already covered this to some extent, but what fraction of mammalian transmembrane proteins are beta barrel proteins, and are there any connections between those and bacterial beta barrel transmembrane proteins? And of all the known beta barrel transmembrane proteins, what fraction is your estimate do not have a known 3D structure? I'm just trying to extrapolate the territory of where this is going.
Yeah, so they're like roughly 2 to 3% of a typical genome should have, a bacterial genome has these transmembrane beta barrel proteins. Then for mammalians, I am not really sure how, I think it's a very, very low number, within hundreds, I would say. And we only know, well, so we focused on mitochondria. At least in mitochondria, we know that there are only maybe 2 or 3 of these transmembrane beta barrels. Yeah, I guess they're also prevalent in chloroplast. So then again, it's roughly in the outer membrane there, and that again is like 2 to 3%.
And plants, do you know?
Chloroplast in the plants.
Okay.
Right.
Okay, so with that, let me thank the speaker again.
Thank you.